are going to introduce some engineering tools like microarrays, micro devices, microfluidic device, high throughput tools to help us for drug discoveries. And we also have some specific topics like novel drugs, modalities like biologics, the one that not what we usually think about drugs. And also we are having one topic about cancer drugs, so specific focus on cancer drugs, which is a big category. And if we have time towards the end of the semester, we're going to cover some other topics as well. Okay, so about the structure of the course. So as I said, I will upload slides after this course. So in the future, I will try to upload it right before we start the class, so you can have some like PowerPoints to write notes on. And also, we will have some assignments, not for every class, but maybe even every two lectures, so total about 6 assignments. And those assignments will count for 25% of the total grade. So they're take-home assignments, so it's not like in-class quizzes, so you have a week to finish them. So they are due. So I will give you assignments, for example, at the end of this lecture, and you will hand them to me at the beginning of the next lecture. All right, so you have a week time to do them. And if we have assignments in the last lecture, we will take about like 30 minutes to talk about them in the next lecture, all right, after you have already handed in your assignments. So I find that very useful because people will have some questions in their assignments. So it's that time, the tutorial time, would be a chance for you to really clarify any confusions you have. So those assignments are all from the contents of the past lectures. So the purpose of them is to help you really digest and understand the concepts. All right, and then we will also have a presentation, which is a group-based. So I will decide how many people in the group after the add and drop period, so I know how many people enrolled. So right now there are about like 45 students enrolled. So probably we will have 2 to 3, or 2 to 4, I'm not sure yet, people in one group. And one group will give one presentation together, and you'll be graded as a group. All right, so yeah, we'll decide on that later. And this presentation will be 25% of the final grade as well. And then the rest of 50% of the grade is from the final exam, which will all come from the contents that we talked about. All right? Any questions so far? Okay, cool. So this is the class schedule. So today will be the lecture 1. We have 2 holidays during the semester, so no class on that 2 weeks. And the group presentation will be covered in the last 2 weeks, so week 10 and week 12. All right, so I will decide on the format as soon as possible and ask you to form a group soon. So you can form a group, and then towards the end of the semester, you will be asked to submit your topics for your presentation. So there's no report, so we just do presentation. So you need to discuss with the group member and make a PowerPoint slides and present in front of the whole class. All right, so that will be the format. Okay, so that is all everything about formality of this course. Any question?
For the group presentation, will it be assigned, or we can make our own group?
You can make your own group, and also you can choose your own topics, as long as it's related to this course.
So each two lectures will provide one assignment.
It's not that regular, so maybe after we finish like one topic with one big topic, I will give a assignment. So in total there are 6, so probably scattered across all lectures. So there'll be no assignments today. So yeah. All right, cool, great. So let's get started. Let's get to the topic of the drug discovery technologies. So before we talked about how drugs can be screened and discovered, we need to understand what is a drug. So what are we talking about when we talk about drug discovery? So what's the definition and what's the classification of drugs? So what is a drug? So now you can take a few seconds by yourself and think about, when you first think of this word drug, what's coming to your mind? So you're probably thinking the medicines that you're taking, right, when you're sick. Like medicines that can help you overcome a cold, overcome a flu, or a headache or something. So actually the definition of the drug is quite broad. It's much broader than just the medicine. So for example, any food or substances or plants surrounding us can be considered as a drug. And the history of the drug is very, very ancient. So people are already starting to taking drugs even before they give it a name, or even before they know why it works or how it works. So for example, here people found, like, the archaeologists found a painting showing that the ancient, I believe this is the ancient Sumerian people. So this drawing showing they are using the willow tree bark, chewing the willow tree bark as a pain reliever. So they don't know why it works, but they somehow, based on everyday experience, they know that will relieve their pain. And also, in this ancient book from Egypt, there's description showing that a wound if a wound is inflamed, you can get the heat out using the leaves of the willow. So you can see the words are very not accurate, like describing the disease as a heat, but those willow leaves or barks, it can also consider as a drug. And of course, opium, we all know that it is a very ancient pain reliever, and people have been using it for thousands of years. And similarly, like the coffee and tea we take every day, they contain caffeine, which is also considered as a drug. Then it comes down to the normal medicine that we take. So like when you have headache, you can take the anti-inflammatory drugs to relieve your pain. And when you have a bacteria infection, you can take antibiotics like penicillin to suppress it. And we have medicines to control your blood pressure, and for the mental health issue, we also have antidepressant medicine. So these are more modern, but I want you to know that the definition of drug itself is actually quite broad. So strictly, any chemical substance that can be used in the treatment, cure, prevention, or the diagnosis of disease, or maybe not disease, you just want to enhance your physical well-being or mental health well-being, like you want to drink coffee to keep awake or keep a spirit. Any substance like that can be called a drug, all right? Okay, so it's a very broad definition. Then the next thing, a very important question we want to ask is, how do drugs work? So we know that when we're sick and we take some specific medicine, we'll get better, but actually it's a very complicated process in between. It's like black box. So how exactly those medicine can achieve this goal? people for hundreds of years. So until recently, until recent 100 years, people don't really know how drug works. And that's because they don't have enough biology knowledge. How the drug works actually requires a lot of fundamental level physiology and biology progress, understanding about that. So what do I mean by this? Is because drugs usually work on specific protein targets. And this concept may sound very simple and intuitive now, but it takes people many years to really get to this point, because proteins are hard to study. People don't know, we humans are composed of cells, and cells are composed of proteins, right? So it's a very modern concept. So if you look at this chart, so individual human beings are in this, in here individuals, and we form families in the society. So when you get sick, you feel it in this individual level. And if many people get sick, it forms a pandemic, right? Like the COVID-19. So it's on society level. And when people are developing drugs, they try to cure on this level. However, the real mechanism of how drug works actually target much smaller than that. So humans are made of organs, tissues, and tissues are made of cells, and cells are made of proteins. And drugs actually work on this protein level to cure whatever is showing up, which is the disease. So this is a concept of a receptor theories. So the drugs can be think of as a ligand, and our protein can be think of as a receptor. So once these two interact with each other, the drug will interfere with how the protein function, and it will change its function, and eventually goes up to this chart, and eventually influence the individual and the society. So that's how the drugs, even though the effect we see is on a large scale, how it works, the mechanism actually is function at this very small protein level. And so that brings us, this concept, or the mathematical, or the chemical description of this concept actually is defined by A.V. Hill. So he's a physical chemistry or a biologist, and he won the Nobel Prize in 1922 for some other work, not for this one, but this receptor theory brought up by him is also a very significant contribution of him to the science society. So, and what's more interesting is that the Hughes equation, which describes the interaction of the receptor theory, it was developed by him during his undergrad students in college. So if you're interested in his story or in this theory in more detail, you can check out his Wikipedia page, which has a very good description on that. All right. So now we understand the drug actually works on a specific target to achieve its goal, and that brings up a very important concept called mechanism of action, MOA. So we will use these words a lot during this course, so I want you to really understand what it means. So the mechanism of action is the specific biochemical interaction through which a drug substance produces its pharmacological effect. So basically, MOA describes how the drug interacts with what specific proteins to realize its intended function. All right? And it's very important for us to understand MOA of a drug to optimize and develop some more novel, some newer and or drugs. Okay, so now we understand how a drug works, like in concept, what types of drugs we usually talking about. So there are many different categories we can divide the drugs. So now I just want to describe this classification. So we can divide it to two categories. One is called small molecule drugs, or the traditional drugs. That is the drugs that we probably 99% of the drugs that you're thinking of belong to this category, like aspirin, or like the medicine for flu, or the antidepressant drugs. So basically, just by name, you can understand small molecule drugs, they are relatively small. So they are all small organic molecules, usually contain several carbons, several oxygen, nitrogen, and hydrogen, and that's it, right? Right now, most of the drugs on the market belongs to this small molecule category, and I believe about since maybe in the 70s or before 1970 and 80s, all drugs are in this category. And so they are small organic molecules that people can manufacture in factories. So usually in these types of drugs, there's a component called API, that is active pharmaceutical ingredient. That is just the molecules that's doing the work. And you usually will see those the drugs that we take usually are in the tablet or in the capsule format, right? And the capsule or the starch surrounding it are called excipient, which they are not active. They don't do the work, but they just helps to keep all the active ingredients together, or helps deliver into your bloodstream, or make it taste better. So that's called excipient. So the API and the excipient together will form the small molecule drug that we take every day. All right, so that is one type, and another type is called biopharmaceuticals, or biologics in short. So that is a relatively new type of drugs developed, maybe used in the recent 50 years. So it's in contrast to the small molecule drugs, you can imagine that it is a big molecule drug. So by big, I mean it's not small organic molecule anymore. So they are usually proteins, or even peptides, monoclonal antibodies, nucleic acid, or even a whole cell or a whole tissue. So anything in those categories can be called a biologics. So you can see these are more complex. So they are only developed recent years with people's technology more advanced. And one typical example of biologics is insulin. So I believe everyone has heard of this, right? So insulin is a protein or an enzyme that's naturally produced by our body. However, some people with type 2 diabetes, they lack this insulin, or they don't have an active insulin. So they have to get it from outside. So that's how companies, pharmaceutical companies, they will produce purified insulin and make it into a drug, into the injection. So people with type 2 diabetes can inject those into their body to help them gain a normal lifestyle. All right. So we will have one specific lecture talking about biologics later, because it is a very advanced and very promising direction of the drug industry. And also there's many already commercialized biologics called monoclonal antibodies, which also is one subcategory of these biologics. So we'll cover the details of those in that lecture. Okay? So here are some pros and cons of small molecule drugs and biologics, so you can understand it in more details. So for small molecule drugs, the advantage is that because they are small, we can use organic chemistry synthesis to synthesize them, so they can be synthesized very easily, and you have many ways to do that. So it's not that costly, and also small molecules they can easily transpass your cell membrane, so it's easy to take orally. You can just eat them. On the contrary, if you look at here, this is the opposite. This is the disadvantage of biologics. So biologics are like proteins or amino acids. They cannot be synthesized easily in factories. So they have to be produced using some biotechnology. So which makes large-scale production challenging and makes it more costly to produce. And also, they cannot be taken orally. So you have to do injection or some other way. Does anyone guess why they cannot be eaten? Can I just eat the biologics and just take it like a pill, and why doesn't work like this? Yes.
It'll get broken down by your GI system.
Exactly. So the protein or the amino acid, they're just like meat, right? So these biologics are no exception. So they will, if you take them in your GI tract, you have enzymes to break down proteins. So once they're broken down, they lose their structure. So they lose their function. So they don't function as you intend them to be. So that's why you cannot simply eat those biologics, but you can inject them directly into your bloodstream. So that won't go through your GI tract. So they will they will keep intact until they reach their final destination and function. All right. So this will create a barrier for delivery, right? So you need a needle, and it's quite painful to do that. Okay. So, and also biologics are usually species specific. So when you develop biologics, you may use animal model to do your experiment to test it. However, even if you get really excellent results, it may not work on human. So that will also create challenges for the development of biologics. Okay, so biologics, what advantage it has? So it is usually a quick discovery process, because when people develop biologics, they already have a target, like a protein target in mind. So, or usually people already naturally produces those kind of substances like insulin. So all you need to do is to think some way to get a lot of them. So, but instead for small molecule, for example, you know the willow tree leaves works, but you don't know what substance in it works. So you have to do a lot of screening, and that can take many, many years to really select out the active molecules. All right, so on the quick discovery process concepts, one excellent example is the COVID-19 vaccine. So COVID-19 vaccine, we know it's the mRNA vaccine, so it can also be counted as a biologic drugs. So we know it works excellently because people can develop it really quick during this pandemic period. So it's much quicker than the traditional virus method. So it has an advantage in that. And also, since biologics, they are naturally proteins, which we already have in our body, so usually it's less toxic. We don't really reject those kind of things. For small molecule, they can be really easily secreted out by a body, so it can only remain in your body for like 12 hours or 24 hours, something like that. So you have to retake the pill every day. So you know that some medicine you need to take it 4 or 5 times a day, right? But biologics can usually last several days or even months, so you don't have to inject that frequent. So that is the pros and cons of these 2 big categories. We'll talk about them in more details later, but you can see that they have their respective limitations and advantages. So you need to choose which one to develop based on your purposes. Okay. So let's see. We now know it's a hard job to screen a drug to find the perfect drugs. And now we're going to talk about some major ways people use that they can use to screen or discover a drug. So the most simple and intuitive one is called a phenotypical screening. So just by the name, you can imagine how it works, right? So it's just by observing the phenomenon, the drugs act on the tissue or on the cells, so you can figure out which one gives you the preferred phenomenon. Most of the drugs in ancient time or up to 100 years ago are discovered in this way, or I would say most first-in-class drugs before 1980s are found in this way. So first-in-class drug means it is the first one in this big category. So many drugs, they can be considered as one category, so they have exactly identical MOA, so all these drugs, they have the same MOA, so we don't consider them all novel drugs. Only the first one in this big category, we call it first-in-class drugs, all right? Okay, so this is the picture of ancient Chinese painting. So this is a book called 本草纲目. Maybe many people already know this. So this is how an ancient character in China, it takes thousands of herbs to find out the useful ones. So it is considered one of the earliest doctors or pharmacologists in China. And he summarizes observation and his conclusion in this book, so to help people which herbs can help you do what things. And this is a very typical phenotypical screening method. So he doesn't know this word, but that's what he's using. Okay, so in the phenotypical screening, what we usually do is we just try many different molecules or compounds and throw it into our target, for example, into our cell cultures, and we just observe the final phenotypes. So maybe the cells will change shape or change color or whatever, and we will test thousands or millions of compounds and select out those that give us the what we want. Okay, so here is a example I want to show you. So those 96 well plates, on each well plate, we have several embryos in it. You can see those, the round circles are, one circle is one embryo. And into this one well, into one well plate, plate well, we add one molecules into it. And so the experiment, the purpose of this experiment is to screen out a molecules that can trigger the muscle action of some proteins. So we will monitor the movement of these embryos like those. We will use a microscope and to monitor how it moves. And if a drug is what we want, if a drug is what we expect, it will show a certain pattern of movement. All right. So with this high throughput monitoring assay, we can automatically using the microscope, the robot, to give us the movement pattern into each well and observe the movement of each embryo simultaneously. So once we saw a specific pattern that we want, we can figure out that compound is what we really want. And then we can do many rounds of this until we select the most effective one. All right. So usually in this phenotypical screening method, we either use cell or here we use embryo, or even we can use a whole animal model. So the advantage of this kind of phenotypical screening is that since we directly work on a very large scale tissue or organ, very likely the screen out molecular compounds will be active towards human as well. So, and it will increase our successful rate for the downstream development. Okay, so that is called phenotypical screening. And then the next method, well, it's not exactly a method, but it's a concept called fast follower approach, also called me-too drugs. Basically, we don't develop a new drug from scratch, but instead, we modify some existing drug, maybe add a functional groups or remove some functional groups, change some atoms, and we make it slightly different. But this slight change probably can increase the property of this drug somewhat in some extent. And we call it analogs of these drugs. And those drugs, after it's developed, we call it the me-too drugs. So instead of a first-in-class drugs, but it's still patentable, so people can still make profit from these me-too drugs. So you may think, oh, this is not interesting, right? This is not novel, like innovations. However, actually, believe it or not, almost 60% of all drugs on the market are me-too drugs. So it's really hard to find a really new drug. So people are really just trying to optimize the existing drugs and make it better. So it's still useful, right? It's just they have the exactly identical MOA than the existing drugs. Also, because it's a profitable for company, and also it's much less costly than developing new drugs from scratch. So big companies really also like this approach a lot. And as I said, a big percentage of drugs on the market belong to these me-too drugs. So advantage of this following approach is that since we already have a working drug in hand, there's much less risk that the drug will fail, and it will have less risk for toxicity of the drug as well. Okay, so next we have a third approach called target-based screening. So if you think of this phenotypical screening, it's a trial and error process. We just try thousands of compounds to figure out which one gives you the good final results. And even if we figure out one effective drug, we still don't know how it works, right? We don't know the MOA of that, but we don't really care in this phenotypical screening. Instead, for target-based screening, it's the opposite direction. We start with a target that we know is important. Like, for example, for type 2 diabetes, we know like certain protein is important, and so we will focus on that protein and find a drug that can directly interact with that protein, and hopefully can change the function of that protein. So that's a target-based screening. So we start with the target and then screen for an active molecule or compounds. All right, so this target-based screening is relatively much recent than the phenotypical screening because you need to know the target. To know the target, we know the target are mostly proteins, right? So to know the target, you need many like high advanced technology like biotechnology or gene technology. And it's only possible for recent years. That's why in recent decades, this method has becoming more and more popular. And about 38% of first-in-class drugs are found in this way. But again, I want to emphasize that it is not necessary to know the exactly target or the exact MOA of a drug before we can use it, before it is safe to be put on the market. A very typical example is aspirin. So aspirin is the active compound that people selected out from the willow tree, and people know it's working, but it's synthesized in 1897, so almost 200 years ago. But the MOA of this molecule is only understood 1970, almost 100 years later, after it's being widely used. All right. So as long as it's approved, it's proven to be effective and completely safe, it's okay to be put on the market without knowing the specific target and the specific MOA. But you may ask, then what's the point of us to study the MOA? So to study MOA and to figure out the target will actually give us a chance to optimize the drugs. So many me-too drugs are developed in this way after we know the target or MOA of a specific existing drug, and then we can really modify the whole chemical structure. So it's still very useful. Okay, so here I'm listing these 3 different types of drug discovery approaches. So you can read it yourself in more details later, and the pros and cons of these 3 different approaches. Like the target-based is we start with a clear target and MOA, so it's we have a clear mind how to optimize it. And on the contrary, the phenotypical one screening, it's more like a blind screening. But also the target-based screening has disadvantage that even though you exactly know the interaction between the ligand and the drug, it may not eventually work on the whole human level. But on the phenotypical screening, since you already start with a very high-level experiment, it's more likely to go to the reaching the ending point. And also the pros and cons of the me-too drugs, which we also just briefly mentioned. Okay, so here is some statistics of these different types of drugs and different types of discovery approach. So you can see here, this is the statistics from 1999 to 2008. I just copied from a book. So you can see the first-in-class drugs are mostly discovered by the phenotypical screening, which makes sense, right? But for the follower drugs, most of them are developed using the target-based screening because after knowing the target, it's very easy and intuitive to do some modification and make some follower drugs. And this is the how many drugs people discovered along the years. You can see there is a slight increase of the first-in-class drugs since it's more difficult to develop. But the follow-up drugs, it has a much higher number and also a much larger increase along the way. So you can, which is expected because the follow-up drugs are much cheaper and easy to develop. And also, we have the small molecule drugs, the conventional one versus the biological one. You can see the biological one are increasing a lot recently as well. And hopefully we can see a much higher percentage of this biologic with the technology being more developed. Okay, so we'll stop here for the first section.